Hi, I'll be presenting the fundamentals in biotechnology. I hope you enjoy the video. As for this presentation, I'll be explaining three main concepts: the central dogma, gene expression, and the tools of synthetic biology. By understanding these notions, you'll be able to relate the different components in synthesis of different materials. Thus, setting aside other presumptions, let's first discuss the basic understanding of a central dogma. What is the central dogma? The central dogma is simply the process of genetic information and cells from DNA to RNA and finally to proteins. Genes first regulate the sequence of mRNA molecules and as a result assembles the protein sequence. In other words, the central dogma enables DNA to form a functional product such as protein. This leads us to our next point. How are we going to make use of transcription and translation? As seen, transcription and translation were major processes in synthetic biology. In simpler terms, transcription is a process that ensures that DNA information of each singular cell is converted into RNA messages. It is when the information in a strand of DNA is then copied into a separate molecule of RNA, the synthesis of RNA from DNA. Like its name suggests, translation is a process in which the sequence of RNA molecules are converted into a sequence of amino acids during the process of protein synthesis. Thus, this concludes the central dogma concept. What is gene expression in simple terms? Gene expression in a singular term would simply be when DNA instructions are converted into products such as protein. It's able to act as a switch when generating protein, as well as the ability to control the volume of protein being made. As shown in the diagram, it is the process in which the encoded information in a gene is used in order to assemble the molecule of a protein. There are a number of different appliances in the synthetic biology toolkit: genome engineering, cellular computation and memory, genetic, circuit induction, biomaterials, riboregulators, and so on. First of all, the most basic element in synthetic biology, genome editing. On a personal outlook, I view genome editing as an assembly of different building blocks being inserted and built on one another. It's when DNA is inserted, modified, or replaced in the genome of an organism. In broader terms, I'm of opinion that synthetic biology will certainly transform our healthcare treatments and even alter the world we see today. Truly a tool that will transform the industries today. Thank you.